Hello everyone. Welcome to the 2014 Bio-Rad ProteOn webinar series. My name is Ruben Luo, and I am the product manager of the Bio-Rad ProteOn XPR36 product line. Today, we are happy to have Dr. Katie Tarun as our webinar speaker. Dr. Tarun obtained her PhD in chemistry from the University of California, Berkeley in 2011, where she studied the biophysics of transmembrane protein translocation. Currently, she is a postdoctoral fellow in clinical chemistry at the University of California, San Francisco, where she has been investigating the use of surface plasmon resonance, or SPR, and high-resolution mass spectrometry in the clinical lab. In particular, her research focuses on clinical toxicology and therapeutic drug monitoring. In today's webinar, she will be telling us about her work on quantitating the TNF-alpha inhibitor drug infliximab in serum using SPR technology. During the webinar, you can type your questions in the Q&A box and send to me. I will hold the questions until the end and pass them to the speaker. Okay, let us welcome Dr. Tarun to present.
Hi, everyone. Thank you, Ruben, for inviting me to speak in this series. And thank you to those of you who are joining me. I'm really excited to tell you about my work on the ProteOn. I hope to give you kind of a general overview of how SPR might be used in the clinical lab and provide also an overview of some of the things that you should think about when working with a complex matrix like serum. So I know many of you are already familiar with surface plasmon resonance, but this is actually not a commonly used technique in the clinical laboratory. So I'd like to briefly explain some background about the technique. So most SPR instruments are set up in the following manner. You have a light source, a glass prism, a metal surface which is usually gold, and a detector. Light is directed through the prism to the bottom of the metal surface, where it gets reflected back to the detector. However, at a specific angle, called the resonance angle, electrons in the metal surface actually absorb the light, and you get an intensity minimum at the detector. So these electrons, also known as surface plasmons, are very sensitive to the environment at the metal surface. So any changes at the surface cause a shift in the angle at which the light is absorbed. So you get a shift of this intensity minimum at the detector, and this is the basis for your SPR signal. So the general SPR experiment is fairly simple in design. You have a chamber that is under constant buffer flow. You're first going to mobilize your biomolecule to the surface, bind Analyte, allow your Analyte to dissociate, and then you wash off any remaining Analyte, and you can repeat this binding cycle. So the data that you get out of these experiments looks like this. So this is the sensogram. So here you have your Analyte injection. You're first going to have Analyte association, followed by dissociation, and then your regeneration or wash step. So what's really nice about SPR is that it's a very versatile platform. So it's versatile in terms of the types of interactions that you can study and the data that you can get out of these experiments. So first, there's a lot of flexibility in the surface chemistries that you can use to immobilize your biomolecule, and you can immobilize proteins, antibodies, DNA, etc. This allows you to study protein-protein interactions, protein-antibody, protein-small molecule, etc. And I'm sure Ruben has many webinars about all these different possibilities here. So the technique is also versatile in terms of the data that you can get out of these experiments. So you can use SPR to measure binding kinetics, binding affinity. You can also use it more qualitatively to get a yes-no binding answer, and you can use it to measure Analyte concentration. So the other advantages of SPR is that it's a real-time measurement, it's label-free, and the surface can be reused. So from a clinical laboratory perspective, I think the biggest advantages of SPR are that it provides a real-time measurement and is a label-free technique. So in terms of it being a real-time continuous measurement, this first means that the results are generated faster. So using SPR, I can have results for a sample in the time that it takes to finish a binding cycle. So for my method, this is about 10 to 15 minutes. So compare this to a technique like ELISA, where developing a plate can take an entire day. So the time that it takes to generate a patient result, or what's called the turnaround time, is a really important consideration in the clinical world. And if we're comparing SPR to ELISA, there are also fewer steps involved with SPR. So you kind of just put in your sample and you get out your result. So this saves a lot of staff time, staff labor, which is another major consideration in the clinical world, because your staff's time is the most expensive part of the lab. So in addition, real-time continuous measurement may be able to pick up interactions that endpoint assays miss. And finally, real-time measurement can provide additional information. So you can get information about binding kinetics and affinity. It kind of remains to be seen how this additional information may be used in clinical diagnostics. But I bet there will be certain applications where, say, the binding affinity could maybe help distinguish between two analytes, for example. So also the fact that SPR is a label-free technique is also an advantage from a clinical lab perspective. Label-free means that method development is easier or simpler because it requires fewer biologicals. So instead of having a sandwich type assay where you need 2 antibodies, you can be able to measure your analyte with just 1. And you don't really have to worry about labeling or if the labels are going to interfere with binding, etc. So because the method development is a little bit easier and the instrument is very versatile, I think SPR could be a great platform for laboratory-developed tests. So what is a laboratory-developed test? The majority of the tests that we run in the clinical lab are commercial tests. So these are methods that have been developed and sold by manufacturers. In contrast to that, laboratory-developed tests, as the name implies, are methods that have been developed and validated by a single laboratory, and they're not really meant to be distributed to other labs or marketed to other labs. This is just, these tests are often called in-house tests or homebrew tests, or laboratory developed tests, LDT for short. So often the lab will use a laboratory developed test when commercial assays are either not yet available, or perhaps there are a few commercial assays available, but they don't work as well as we want them to. So we will develop methods in-house. And I think SPR could be a great platform for that. Okay, so I'm using SPR to measure the concentration of infliximab in serum. So infliximab is a chimeric monoclonal antibody that targets TNF-alpha. It's used to treat a variety of chronic autoimmune conditions like Crohn's disease or rheumatoid arthritis. However, not everyone responds well to treatment, and this can be due to a variety of reasons. So the patient could be receiving an inadequate dose, or maybe isn't dosed frequently enough. There could be variations in drug metabolism. And finally, patients also tend to develop antibodies to infliximab over time, which reduces drug efficacy. So therefore, it's really important to be able to measure infliximab levels in serum to distinguish between these different reasons and better manage the patient. Over the last decade or so, several studies have tried to correlate serum concentrations of infliximab to drug efficacy. And in general, they've found that infliximab concentrations greater than 12 micrograms per ml at 4 weeks after dose, or concentrations greater than 1 microgram per ml at trough levels so trough levels is sampling right before the next dose. These concentrations correlated better with better symptom relief. I should say here that this is kind of still an active area of research, and the infliximab reference ranges are likely to vary depending on the specific disease state that infliximab is being used to treat. But in general, we're looking to measure infliximab concentrations in this range. So currently, there are 3 reference labs in the U.S. that offer quantitative tests for infliximab. However, all of these tests are very expensive, and this often limits how often we can order those tests. So in particular, I work at a city hospital that works on a fixed budget, and we really can't afford to send out many of these infliximab tests. So we were actually approached by a GI specialist in our hospital who was wondering if there was any way that we can develop a method for this in-house, and we thought this would be a great application for SPR. Okay, so when developing a method for SPR, some of the earliest decisions that have to be made are what surface chemistry are you going to use, and what are you going to put on your surface. So I decided to use a GLC chip, which uses amine coupling. So this is a covalent linkage to the protein. I chose it because it's kind of the generic surface chemistry, gives you a medium amount of immobilization. It's kind of a great place to start when developing a method. So I'm using human TNF, immobilizing human TNF alpha to the surface. This was purchased from a company. It's recombinant. And for our infliximab, we were able to obtain leftover infliximab from a patient's treatment. So for method development, especially the earlier stages of method development here, I'm going to be spiking infliximab into control serum. So this was just normal serum from a healthy volunteer. And because I'm working with serum, which is a fairly dirty, complex matrix, there's the potential for nonspecific binding. And you kind of expect some nonspecific binding here with serum. But there are several ways to minimize that or to correct for it. And one of the ways that I'm trying to minimize nonspecific binding is I'm using a lot of detergent in my buffer. So I'm using 0.05% tween, and I'm also including a lot of bovine serum albumin in the running buffer as a way to coat the surface and help reduce nonspecific binding. Okay, so once some of those early major decisions are made about the method, it's time to optimize conditions. So in developing method, you really have to optimize each step of the binding cycle here, so the mobilization, analyte interaction, and the regeneration step. I'm not going to spend a ton of time going through the details here because every method is going to be different, and this is kind of a straightforward process since most of these parameters are decided empirically. So it's really just about cycling through the different conditions to see what works best. The really nice thing about the ProteOn is the 6x6 grid design. So it makes this process go a little bit faster. So there are 6 lanes created vertically, and you generally immobilize your protein to the surface in this direction. And then there are 6 channels that are created horizontally where you're going to inject your analyte. So you essentially have 36 different spots on your chips, and if you wanted to, you could study up to 36 different ligand analyte combinations, or you could use this in a way to cycle through a variety of different conditions very quickly. And I found this to be really convenient. And at this stage of the process, I kind of had a couple questions or goal in the back of my head. First, I wanted to make sure that ligand immobilization down a column was going to be consistent. So ideally, how I wanted to use the ProteOn was to inject 6 different calibrators across the chip surface and then use an entire column of data as my calibration curve. So in order to do this, you want to have a pretty consistent surface down a column. So another thing that I was thinking about was how would I be able to minimize or handle nonspecific binding. can I see clinically relevant concentrations, and is the signal going to be reproducible? So when you wash the surface between binding events, are you going to be able to maintain enough signal, fairly reproducible signal over time? So for the mobilization step, this is what the raw data looks like for the TNF-alpha mobilization. There are a couple different parts to this. So first, I'm showing you data from an entire column. So there's actually 6 different traces here overlaid. And so you're first going to activate your surface. And here you're creating carboxyl groups on the surface, which then react with free amines on your protein. And then you have your deactivation step where you're basically blocking any unreacted carboxyl groups. So for the mobilization step, there are a variety of parameters that you can play around with to optimize conditions. But ultimately, the goal here is to figure out what is the optimal ligand density, how much protein should you be putting on the surface, and what conditions work best to get that density. So again, there's several things that you can play around with. You can change how strongly you activate the surface or how long you activate the surface. Here, I decided to use a pretty standard conditions for the activation step. And what I decided to focus on instead was the conditions for the protein. So with the protein, you can change the concentration. You can also play around with buffer conditions of the protein. And probably the most critical part there is the pH. So when you create carboxyl groups on the chip surface, it makes the surface negatively charged. So you want your protein to be positively charged so it's attracted to the surface where it can react with the carboxyl groups. So you want to be working at a pH about 1 unit below the isoelectric point of your protein. Since TNF-alpha has an isoelectric point of 6 to 6.5, I was aiming for buffer conditions starting around pH 5. So I actually tested out several different pHs, and here's some of the data. So on the left-hand side, you can see that the immobilization step looks different depending on what pH was used. Here, I'm holding the TNF-alpha concentration constant and just varying the pH. And on the right, you can see the bar graph, which is the final ligand density, or the signal that I'm getting after immobilizing TNF-alpha to the surface, plotted versus pH. You can see that I get more immobilization at pH 5. So what about protein concentration? Here I'm holding the pH constant at pH 5 and varied the TNF-alpha concentration. And not surprisingly, the higher the TNF-alpha concentration, the more protein you get on the surface, although immobilization does appear to be leveling out around 4,000 RUs. And more ligand on the surface is not always better. So ultimately, at the end of the day, what you're worried about is the signal when you inject Analyte. So here I'm showing you the signal from my 20 microgram per mil infliximab calibrator. And you can see it kind of tops off at around 800 RUs. So this doesn't really improve by adding more TNF-alpha to the surface. So this is most likely due to steric hindrance. Infliximab is a really large molecule. It's an antibody. So it may not be able to access all of the TNF-alpha on the surface, which is why the higher ligand densities aren't really improving the signal. So it's something to think about. So overall, what has worked best for me so far in terms of the mobilization step, I'm aiming for ligand density, so the TNF-alpha density, around 2,000 to 2,500 RU. My protein TNF-alpha is at a pH of 5.0, 5 micrograms per mil. I inject for 200 seconds at a flow rate of 35 microliters per minute. And overall, I found that the mobilization down a column is very consistent. So the CVs ranged from 1 to 5% down a column, and that was really great to see. Okay, so moving on to the Analyte interaction step, the things that I considered most important for this part of the process were what serum concentration should I be using, what is the best way to handle nonspecific binding, importantly, can I see clinically relevant concentrations of infliximab, and as a reminder, we're looking to measure concentrations around 1 microgram per mil at trough, or 12 micrograms per mil at 4 weeks after dose. So I decided to make my calibrators range from 0.3 to 20 micrograms per mil. So I should say a brief word about mass transport, because this is a pretty important concept when doing concentration analysis. So the binding that you observe in SPR is actually a two-step process. So you first have Analyte in the bulk solvent diffusing to the surface, and then your Analyte binds to your ligand. So the process where the Analyte moves from the bulk solvent to the surface is called mass transport. And generally when you're measuring binding kinetics, you want your binding step to be rate limiting, and therefore you want to try to minimize mass transport. However, for concentration analysis, you actually want to work under conditions where mass transport is your rate limiting step. So you actually want to favor mass transport here. Here, then, the initial binding rate that you observe is directly proportional to concentration. So in order to make mass transport limiting, what you do is you actually slow down the flow rate during your sample injection. Another thing that you can do is increase the ligand density on your surface, so the actual binding event is very fast once the Analyte diffuses to the surface. I'm using a very slow flow rate when injecting the samples. I actually use 25 microliters per minute for the sample injection. Okay, so here is what the raw data looks like for my calibration curve injection. First off, I want you to focus on the grid on the left and the middle graph. So I'm showing you data for just 2 ligand channels. So in blue is the ligand channel where I've immobilized TNF-alpha, and the orange is a channel that I intentionally left blank. So I activated and deactivated the surface like normal, but instead of injecting TNF-alpha, I just injected buffer. So it's just a blank surface. So if you focus on the middle graph, there are a couple things that I want to point out. So first, there's this sharp jump in the signal, and that's actually an artifact. It's due to the fact that the refractive index of my sample does not quite match the refractive index of my running buffer. So you try to match them as closely as possible, but it's not perfect, and it can be especially difficult when working with serum because there's so much protein in serum. So the other thing that I want to point out in the middle graph is that if you look at the orange traces, which is binding to that no ligand channel, you can see that there's a slight increase in signal over time. So this is telling me that I do have some nonspecific binding to just the blank surface. What about nonspecific binding to TNF-alpha? So there's a lot of protein in serum, and you'd worry about maybe some of that could nonspecifically bind to the TNF. So the way that I tested for that was to inject blank serum across all rows. And again, I'm going to be showing you data from 2 ligand columns. So here, the middle graph on the bottom, you notice 2 things. You still get a slight increase in signal over time. So there's a little bit of nonspecific binding to the TNF-alpha. So there are many ways to reference the data and correct for this nonspecific binding. And if you focus on the 2 middle graphs, I'm first going to be referencing the blue data to the orange. The result is the 2 graphs on the right. This referencing corrects for nonspecific binding to the surface. And then I'm going to reference the blue data on the top right to the blue data on the bottom right. And this corrects for nonspecific binding to TNF-alpha. So the final reference data looks like this. The nice thing about this is that I can see all of my calibrators. So the signal for my lowest calibrator, yes, it is low. But remember that this is reference data. So this is signal over background. And this is data for 10% serum. So I did try multiple serum concentrations to see what worked best. 5% was really just too low of a signal. 20% does work as well. You get a little bit more noise, more background, because you also have other proteins in the serum at higher concentration. So I decided to go with the 10% serum, and I can see my lowest calibrator at 10%. So I should mention that there's another option for referencing. It's called the interspot. This is actually a unique referencing method to the ProteOn system because of the 6x6 grid design. So the interspot is the space in between the ligand Analyte spots on your chip. So this area of the chip has never seen ligand, and in fact it's unusual too in that this chip surface hasn't even seen the activation or deactivation chemicals that you inject during your immobilization step. So it's a blank surface, but it's slightly different than the blank ligand channel that I just talked about in a couple slides ago. So unfortunately, the interspot referencing didn't work well for me. So shown here, the interspot data is shown in the dotted lines, and the solid lines are signals from my blank ligand channel. So when the calibrators are referenced to the interspot, you actually get some negative signals because now you're overcorrecting here. You can see this a little in the bottom graph. So I'm getting a little bit better binding to the interspot than I am to just my blank ligand channel, which is why I can't use the interspot for referencing. Okay, so finally the regeneration step. So the idea here is to wash off any analyte that hasn't diffused away. You want the condition to be harsh enough that you get off all of your analyte, but you don't want it to be too harsh that you're actually going to be damaging the protein that you've immobilized to the surface. So it's a little bit of a balance here. And again, this is a condition that's decided empirically. Typically, the regeneration solution is going to be a mild acid or base. You can also try high salt solutions to disrupt binding, or maybe a detergent like SDS. I did cycle through all the different regeneration conditions, and what ended up working best for me was actually a fairly harsh regenerator. So I ended up using 10 millimolar HCl to wash off the infliximab. So this is what the raw data looks like. You first have your analyte binding and then your dissociation, and really not much dissociates, which makes sense because as an antibody therapeutic, infliximab is really designed to bind tightly and stably to its target, so not much comes off. And so the sharp drop in signal that you see is the injection of the 10 millimolar hydrochloric acid. And what I want to point out is that what you're aiming for here is to be able to wash off all the material and restore your signal back to baseline, which you do. So one concern that I had was, how is my signal going to hold up? So I'm using such a harsh regenerator. Will the protein on my surface hold up? Is the signal going to be reproducible over time? So here I'm showing you data from a single analyte ligand spot on the chip. And these are 2 injection wash cycles of where I'm injecting infliximab. These are the same concentrations. First, you can see that I'm restoring the signal back to baseline after washing. And second, the intensity of the infliximab binding is approximately the same. So in terms of the long-term reproducibility, here I'm showing you multiple injections of my 20 microgram per mil infliximab calibrator. and again, it's for a single Analyte ligand spot on the chip. The time scale here is a little bit deceptive because of the way the software concatenates the data, but this is actually data collected over about a week, and there were also many injections in between the ones that I'm showing you here. So the nice things out of this data was that it's really reassuring to see that I'm able to maintain approximately the same signal after repeated wash signals and maintain a signal over time. So there is some variation in the signal, however, and because of that, I'm going to have to normalize the data when I make a calibration curve. So in order to make a calibration curve, I'm going back to the reference data for my calibrator. So you've already seen this graph on the top. You actually just take the initial binding rate of each calibrator. So you're measuring the slope of the line at the very beginning of the binding step, and then you plot the slope versus concentration. So again, because the signal varies slightly between injections, I decided to normalize all of these rates to the rate of my 20 microgram per mil calibrator. So it turns out that the normalized calibration curves are very consistent. So if you look at the graph on the left, I'm showing you about 8 data points here overlaid. So this curve ends up being very consistent. This is data across multiple injections across different ligand columns. So that was great to see. That's very reproducible when normalized. When I take all of the calibration data that I ran over the course of this chip, I averaged those data points and fit this to a 4-parameter logistic equation. So that graph is shown on the right. Using this calibration fit, I went back and back calculated all of the known concentration samples that I ran across the chip. I then compared these back calculated values to the concentration that was expected in order to calculate a relative error. So here the relative error is basically the relative difference between the calculated value using my calibration curve and the expected concentration. So this is a graph of what I'm getting for the relative error, and obviously you can see that there's a lot of scatter, especially when you go to the lower concentrations. But what is reassuring here is that there's no bias. So all of the points are kind of evenly scattered about 0, meaning that there's no bias in how I'm fitting the data to make my calibration curve. So ideally, you want the relative error to be under 20%. I'm kind of close to that around my 1 microgram per mil calibrator, but it certainly falls off at the lower concentrations. This is really an effect of trying to measure concentrations in the flat part of my calibration curve. So if we go back to my calibration curve, I'm trying to measure those concentrations below 1 microgram per mil is kind of the flat part of that curve. So any variation in signal down there is going to lead to a lot of error in the concentration. In terms of the precision of the data, this is what I'm getting for my CVs. And I have about a functional sensitivity, which means a CV of 20% at about 1 microgram per mil. So currently, the performance of our assay would work well if we're measuring infliximab concentrations 5 micrograms per mil and above. So we could use this to measure patients that are drawn midway between doses, but we would certainly have to improve the sensitivity if we're going to be measuring trough samples, which we would expect to be around 1 microgram per mil. So we were able to obtain 10 patient samples. Most of the patients at our hospital have blood work done right before their next infliximab infusion. So most of these samples that I'm showing here in fact, all the ones on the left, top left here are trough samples. So the traces in blue are my calibrators. So the one on top is my 20 microgram per mil calibrator, and on the bottom is my 1 microgram per mil calibrator, and the patients are shown in orange. So you can see that most of the patients are running at or below my 1 microgram per mil calibrator, although there are a couple that are in between. So we were also able to obtain a sample from a patient who had their blood drawn 3 days after their infliximab dose. So this response is actually really high. It's way higher than my 20 microgram per mil calibrator. And when calculated out using my calibration curve, the graph on the bottom is showing you what those values come out to be for these patients. Most of the values, again, are around 1 microgram per mil or lower. And this is the patient 3 days after dose. Interestingly, this patient sample was sent out to a reference lab for measurement, and the reference lab reported the sample to be less than 1 microgram per mil, which is their cutoff. So that was nice to see because that correlates well with our measurements. Unfortunately, it's been very difficult to come by a reference method to compare our results. As I mentioned earlier, pretty much all the reference methods are very expensive. But recently, we are going to be collaborating with a major reference lab. They'll be helping us out and sending us samples so we can compare our method to. I was hoping to have that reference data to show for this presentation, but unfortunately it just didn't work out. But overall, the patient data is promising, however, because we're getting values that are appropriate levels for trough samples, and we are seeing a dose response for the patient that was sampled 3 days after dose. So this was really promising to see. We are able to measure infliximab in patient serum disease matrix. So to summarize, developed a quantitative method for infliximab in serum. It's certainly still a work in progress, and we are close to having reference values for our method to see how well it's working. We hope to improve the sensitivity so we can be able to measure trough samples better. Still need to investigate and are in the process of investigating potential interferences in disease matrix, and we also hope to develop a way to detect antibodies to infliximab. But overall, this has been a really fun project to work on, very interesting. The ProteOn is easy to use, and I think it has great potential for clinical applications, research applications, and it's been a really great project to work on. So with that, I would like to thank the people at Bio-Rad. Bio-Rad has provided funding for this project. I'd especially like to thank Gary Ross, who's been very helpful. He's the application scientist. And I'd like to thank my colleagues and advisors at UCSF. And thank you for your attention. I'd be happy to take questions.